Please welcome the Atlantic CEO, Nicholas Thompson.
Hello, I'm back. Thank you all. I hope you all had a wonderful lunch. I hope you're ready for the rest of the programming for the day, which should be excellent. I want to start here by thanking Eli Lilly, sponsor of the next round of conversations. Without our sponsors, no Atlantic Festival. We are very grateful. Second thing I'm going to do is I'm going to do another one of those little videos where I kind of break into what my wife calls my TV character. For those of you who weren't here this morning, for the last 7 years, pretty much every day, I've posted a video about something that's interested me in technology policy. We're filming them here, so here we go. The most interesting thing in tech, yesterday I had the opportunity to have a conversation with Jessica Rosenworcel, the head of the FCC, Lena Khan, the head of the FTC, and Rohit Chopra, the Consumer Finance Protection Board. And I asked them, I said, as we head into the era of AI, if you could be, as one of the presidential candidates said, I can't remember which one, if you could be dictator for a day, what would you do? And they had some pretty interesting answers. They had two frameworks to synthesize a little bit and compress a little bit, one of which I think most everybody would agree with, and the other pretty darn controversial. So the first one came first from Rosenworcel. And she said transparency. We need to have much more transparency in the age of AI. You need to understand where the algorithms are working and what they're doing. If you are conversing with a bot, or you have a voice in a robocall, you need to know if it's AI or if it's a human. You need to be able to know if you are talking to a bot or a person for democracy to function. This is something that Chopra agreed upon and something that Khan agreed upon too. You need to know where the algorithms are working. You need to understand where companies have put in AI algorithms. You can understand if there's algorithmic collusion. You need to be able to understand what the algorithms have done. So in case something bad happens, you can trace it back. That was principle one, transparency. I think most people agree on that. It's a good principle. Principle two, and this came from Khan and Chopra, not so much from Rosenworcel. We have lived since roughly 1994 with a regulatory framework in this country where the platforms build the tech and it's kind of treated as pipes. And if something bad happens on a platform or because of a platform, it is mostly the person who did the bad thing's fault. It is not the platform's fault. It's kind of the underlying logic of Section 230 of the Telecommunications Act. We have been arguing about that in different ways. There are all kinds of court cases. There's one that just happened in Philadelphia involving TikTok, which is pretty complicated. We have been arguing about that since. But what they were suggesting is that the burden needs to maybe be flipped in the age of AI. Well, maybe if something bad happens because you built an AI and somebody used it for a bad thing, maybe the responsibility is on the company that made the AI. Or maybe the companies that made the AI should have a requirement that they should think through all the possible bad things and build against it. That would be a very different regulatory framework. That would be an extremely different way of thinking of AI. It could be quite frightening. It could be quite bad. One of the reasons all the big tech companies are here and not elsewhere in the world is because they've had regulatory protection, but maybe in the age of AI, the rules need to work differently. It's a pretty provocative argument, and it was the most interesting thing in tech.
Here to discuss the science of aging, please welcome Celine Halioua, founder and CEO of Loyal, Cynthia Kenyon, Vice President of Aging Research at Calico Life Sciences, with Ross Anderson, staff writer at The Atlantic.
Okay. Cynthia, Celine, I'm so glad that you guys are here. I'm really excited about this conversation as a person who is aging and who has a dog also.
Is
Cynthia,
your dog aging?
What's that?
Is your dog aging?
You know, undetectably, but yes. So, Cynthia, I want to start with you, and I was hoping that you could take us back. Now, I just want to flatter you for the audience here so they have some idea of the tremendous contributions you've made to this field. Cynthia was the first person to double the lifespan of a very tiny organism called a roundworm. Not that we want roundworms living forever. But nonetheless, a huge step in this field. And I want to talk about that in a little bit. But first, tell me what this field was like before that.
Oh my God, yeah. So, well, understandably, as you age, you sort of fall apart in different ways. Like an old car, it's a lot of entropy, etc. So people generally thought that aging is just something that happens, and there's nothing you can do about it. There's nothing you can do about it, except maybe you could exercise and be a little healthier. But fundamentally, it's just a process of deterioration. And so there was a lot of resistance to it. There were people in the field working. This was in the early 1990s, a long time ago now. But molecular biologists, I was at the University of California in San Francisco, and molecular biologists there, I mean, it was just a sort of a cesspool, really, of science. People thought, well, you'd work on aging. First of all, if you have delusions of grandeur, number one, and number two, you really weren't good at anything else, so you would go and do this. Seriously, so it was really hard to get any graduate students to work on it, really, really hard. Because people thought, they also thought, if you look in nature, you can see that there are genes for aging because dogs age faster than people. And if you look all around at different animals, they have very different rates of aging and lifespans. And that's because they have different genes. And the reason they have these different lifespans is because gene changes during evolution gave them different lifespans. So right off the bat, you don't have to do any experiments. You know that genes control aging. But even there, people will think, well, there'll be all sorts of genes, many for the liver, some for the skin, and they'll all have tiny little effects, and so you never find them. So it was just negative, negative, negative.
And the work you did, it was just a single gene mutation, and I'd love for you to describe how you did that, but also this was before email, but maybe your snail mail, your phone calls, you must have had a lot of people reaching out to you, and maybe some weird people too.
Well, I'll just talk about this story because it's a really cool story first. So basically, I happened to already be studying this little teeny weeny roundworm about the size of a comma in a sentence called C. elegans. And I was studying, how the fertilized egg creates a nice animal with all its cells and everything. But these little guys age and die in just a little over 2 weeks, and they really get old. So when you work on these animals, what you can do is you can just randomly change genes. I won't go into how you do it, but you can treat them with something called a mutagen, which just changes genes randomly. And then you can ask, okay, do I find genes that affect, muscle development or something like that? Or you could ask, can I find genes that when you change them, make the animal live longer? And it's great because if there aren't genes like that, like if all genes have teeny little effects, and there's one for the head and blah, 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 then you're not going to ever find anything, so you can just stop. Easily, you can just stop. But if there is something, then you have a gene change, and so molecular biology lets you follow it to the biochemistry, to the underlying truth of the system. So we did that. We looked for long-lived worms, essentially, and we were very lucky. And the reason I thought that all these evolutionary biologists were maybe wrong is because in the study, it is a little bit technical, but it's really cool. In the study of the fertilized egg turning into an animal, which is called pattern formation, it turns out that there are gene changes that you can make where one little gene change can cause a fruit fly to have, instead of 2 wings, 4 wings. Like just one change, a whole new wing. Or the antenna turns into a leg. So it turns out, I know, but it's true. But once you get that, once your mind gets that you can do that, which is hard to do, but once you get it, then you think, all these lifespans in nature that are so different, maybe they're, I thought, well, maybe there's kind of a thermostat, sort of. You could turn it up or down. There would be a universal way of controlling the aging rate, and it's set to run fast in the little worm, and it's set to run slower in the dog and really slow in humans, for example. So that's the idea I had based on these extra wing ideas. So, because I used to study that. So anyway, you just need a mission. You need sort of something that motivates you, and that motivated me. So we found them. We found mutants that is an animal with a gene change that lived twice as long as normal, and it was amazing because you look at these little worms on their little culture dishes. They look on it's something like Jell-O. They're eating that with bacteria on it, which they eat. Anyway, so you'll see the normal worms, after about 2 weeks, they're either dead or they're almost dead. They look horrible. And then at the same time, you see the worms, the long-lived worms, they look young. So I have a nice story about this. What if you are, you're in your 40s and you're dating. Say you're a guy and you're dating. and you go out with a woman, let's say, and you're having dinner with her, and you like her, and you ask her how old she is, and she says, and she thinks she's your age, and she says, I'm 80. And I know, that's what it's like. So you look at these worms, and they, seriously, I'm really
Ross,
serious.
you look triggered.
They look younger. It's not like those pictures that you see of people that look 90 that are great. They're running a marathon or they're jumping high, but they still look 90. This is different. This would be a 90-year-old that you think is 45. So it's really like your hairs stand up. Mine are starting to stand up right now. But anyway, that's what we found. And so let me just be sure here I haven't forgotten anything. Yes, so the basic point, that the thing that was really new was that the whole idea that aging just happened and there's nothing you can do about it went out the window. Because what that said is that aging is controlled by the genes. There are genes that actually really control the rate of aging, and that it's plastic. The worms have a normal lifespan, but they don't have to have that lifespan. They could have a twice as long lifespan and age twice as slowly. And so it changed everything, actually. And I can tell you about why they live longer if you ask me the question, or not.
It's very helpful, actually. Celine, I want to ask you, so you have this company, Loyal, that you founded that really is trying to do life extension for dogs. And I was wondering, as I said earlier, I have a dog, and I hope that he lives a very long time. Wish he would live forever. Tell me why dogs for you? Were you like a dog lover, or was it just like a McKinsey thing where it's like, we've identified a huge market? Or
I'm so offended right now. I would like to clarify that I did not get into McKinsey. I failed all the little practice, how many windows are there in New York City thing. And you can still be a successful founder. So I started with dogs actually incidentally, and actually my story is very tied to Cynthia's work. So Cynthia, one of her mentors was this woman named Laura Deming, and Laura Deming was my mentor when I decided, screw university, I'm going to drop out and move to Silicon Valley and go do SF stuff. And she had learned about the biology of what Cynthia was working on, and specifically, I won't steal your thunder on the biology of what you're working on, but the high level, that basically this genetic pathway in worms that more than doubled their lifespan. And since that study had been done, I think it was the late 80s, early 90s, that had been replicated and expanded all the way from worms to mice to rats. There was really interesting correlation data actually in Ashkenazi Jew centenarians having that same or similar genetic change that Cynthia did in the worms, showing lifespan extension, obviously correlation in human studies. And so Laura, when I was 22 or 23, charged me with saying, this biology is so fascinating, and it's been so well validated that if you modulate this pathway in everything from a tiny worm to theoretically a human, that you could extend lifespan significantly. But everybody thinks you can't get a drug FDA approved for a lifespan extension, right? So figure out, how can we take Cynthia's work and turn it into an FDA approved drug for lifespan extension? And of course we started in humans, because that's what I was, a human bio person, Laura's a human bio person. But while I was doing that and I was perusing the literature, I found these really interesting papers looking at the biology of dog lifespan and dog size. So the bigger a dog is, the shorter their average lifespan is. And at the extremes, it's actually a 2x differential. So if you have a Great Dane, they might have a lifespan of 7 to 8 years. While if you have a Chihuahua, they might have a lifespan of 17 to 19 years, right? They live much, much longer. And as far as I'm aware, there's no other species where you see that 2x differential. Don't let the shoes fool you. I'm actually pretty short. I'm not going to live twice as long as some of the taller people here, right? And long story short, it looks like it boiled down to, when we selectively bred dogs to be really big or really small, we selected inadvertently for these genes that Cynthia had discovered, I think 30 years ago at that point. And it seems like we gave these big dogs an accelerated aging disorder. So a Great Dane is literally aging at a 2x faster rate than a Chihuahua. So I thought this was really cool. I've always been a dog person. I grew up with 15 cats, 4 dogs, rescued squirrels. It was a whole thing. But I didn't think it was a company. I never thought I'd start a company. And long story short, I don't know, it was such an earworm. And I remember when I first started thinking about Loyal and telling people about it in 2018, 2019, people would literally laugh in my face when I said the phrase dog longevity.
Yeah.
But I just couldn't let go of it. And it just, people started hearing about it because I would just like talk about it because I didn't have anything else to talk about. So I talked about what I was working on. And long story short, realized that this was an opportunity not only scientifically, but also commercially, which if you're not independently wealthy, which I sure as hell am not, that's actually one of the best ways to develop, new things, right? Can you align commercial incentives with the scientific incentives? And I think that's something we've done really well at Loyal.
And is it, just to connect the idea, is it the case that it'll be easier to get FDA approval potentially in a dog rather than for a human treatment?
Yeah, so easier, but not in the way that people think. So dogs are FDA regulated, dog drugs are FDA regulated. The bar is actually extremely similar to human drugs, in part because people will take veterinary drugs. We all remember the COVID-19 horse dewormer fiasco, which we're both horse people actually, and every time we go and buy dewormer for our horses now, there's like big warnings, like not for human consumption, does not cure COVID, which it does not. But it's quote unquote easier because dogs live such a shorter life. Right? If I'm trying to do a lifespan study in us, it would take decades to see if a drug was having any benefit. And proving the counterfactual is extremely difficult. In a dog, we're running, as far as we know, the largest animal health pivotal study that's ever been run. It's about 1,000 dogs across 60 veterinary sites across the U.S. And we don't know for sure because nobody's done it before, but we think the study will be about 5 years to hopefully show a reduction in mortality risk in dogs treated with our drug. And that makes it feasible just from a commercial standpoint, right? To fund that study.
I imagine that, I don't want to speak for our audience, which may be more scientifically sophisticated than me, but I imagine the exact technical particulars of how this works in the biology of an animal are quite complicated. But could you tell me why, if this has been done in roundworms and it's been done in mice, which are mammals, why should it be that much more difficult to do in a dog? Is a dog like a massively more complicated organism than a mouse?
So maybe we should talk a little bit about the biology. It's really pretty simple.
Oh, great.
So in these little worms, we found out what the gene was that we cloned, that we changed. And it turns out the gene is what they call a hormone receptor. And you all know what hormones are. There's all sorts of them. Adrenaline, testosterone, estrogen, many of them. But the way that the tissues know the hormone is there is they have these things that look like baseball gloves on there that stick out that are called hormone receptors that bind to the hormone. And that's what this gene called DAF2 was, is a hormone receptor. And so what happened, strangely enough, was that when we made the change, the gene change that made them live longer, we made the receptor not work as well. So we damaged it. It still worked because it turns out if you don't have any, you die. Humans die, everybody dies. But if you have a little bit, you live long. And you can ask, well, why is that? So the hormones that are relevant here are hormones that you've all heard of. They're insulin and another hormone called insulin-like growth factor, or IGF-1. Everybody has them. And they allow your body to take up and process nutrients. And they also promote growth. And during the happy stage, when food is everywhere, things are working really well. It turns out, of course, things in the environment can create stresses like less food, or other kinds of stresses like high temperatures or ultraviolet light, things like this, many different kinds of stresses. And it turns out that this is really cool, that having a receptor that doesn't work as well has informational quality. It sort of tells the animal that it might be in trouble. And the animal has a way of responding to that trouble. It turns out these animals could be way more resilient than they normally are. So they turn on all these genes, more than 100 genes in the DNA get switched on. And these genes allow the proteins to fold better. They get rid of damaged proteins better, easier. They prevent infections. They do all sorts of things. There's even something called a recycling station called autophagy in the cells, and it revs up autophagy. So you get rid of all the bad stuff and make more goods. Anyway, so all that gets turned on. So basically these little worms, and not just worms, flies, mice, probably humans, they have in them the ability to be more resilient than they need to be. But why be more resilient all the time if you don't need to be? So probably it's just activated by changes in the environment where you want to not die until times get better. Like if there's not a lot of food, you don't want to starve to death. So you become more resilient, have fewer progeny, and these are what the animals do. And then when food comes back, it's fine. So that's the hormone system, and it's conserved. The animals are resistant to everything you can imagine. It's true of the worms, the flies, the mice, maybe the dogs. She was telling me that her dogs seem sort of fragile, and that the old dogs as they age. But anyway, so that's the biology. It's a very universal biology, and I think it can explain. I mean, I think they live longer because they're more resilient, because the damage of aging, if you will, can be rectified, prevented and rectified. Yeah.
So potentially then there's no difference between a mouse and a dog and how they would respond to this kind of treatment.
Yeah, in fact, we found another gene number 2, that was totally required for the long-lived animals to live long. It's a gene called FOXO, and it's a gene regulator. So it goes sits on the DNA, which are genes, and switches them on and off. And if you don't have that guy, then whether you have this baseball glove change or not, doesn't matter, they don't live long. And it was just shown by someone that that same gene is needed in mice that have this long-lived mutation in order for them to live long. So it's a kind of resilience gene. So it probably really is the same biology. And it might work in humans. So with humans, though, to do a trial, the way it works right now, it might work differently someday. Maybe there'll be a frailty scale or something like that, so you could actually measure aging as it's taking place. People are trying to figure out how to do that. But meanwhile, a lot of this biology, or the biology that controls aging, there are certain diseases where that biology can be like way out of whack, like way too strong. So you'll get cancer, for example, would be an example. In fact, all of these gene changes make all these animals less sensitive to cancer. That's just an example, but it could be other things. So you could imagine that you have a piece of biology and you have a drug that you think is going to turn it down, and it would be perfect to extend lifespan, let's say in a mouse, maybe a person. But what you do is you give it to a person that has a disease where it's way too high. So you just want to turn it down somewhat. And kind of like Celine's dogs, you could almost argue, well, I don't know, that's the difference between aging and a disease is whatever it is. But anyway, these dogs have like way too much of this stuff. So she's kind of bringing it back to normal. It's kind of like as if they have a disease, even though they're nice, happy dogs, they just age faster.
Yeah.
Does that make sense?
Yeah.
I'm going to be
just
quiet.
nod and smile,
That's a good strategy.
Yeah.
Also for dating.
I'm married, so none of these dating jokes apply to me. Let me ask you, and Celine, we'll use the example of dogs. When people think about life extension, the life extension that maybe we don't want is like, you get another 10 years or 20 years of being 90 years old, right?
Yeah.
And with all the kind of, or some of the limitations that comes with. In the fullness of time, if like the greatest vision for Loyal were to come true, would it be the dogs, would they have their prime extended?
Yeah, that's exactly how we think about it. We're trying to extend the healthy middle years of your dog, so the years where maybe you're not going to have that like crazy puppy exuberance, which probably is a good thing, honestly, for your shoes. But your dog, wants to go and play fetch and go to the park and run on the beach, right? And has like the stamina and ability to do that. It's something that we look at in our trials too. So the predominant thing we look at is lifespan extension, in part actually for a reason that Cynthia said, which is just you can quantify it very objectively. The dog is alive, the dog is dead, right? And it's a bit grim, but it's extremely objective. And it's kind of a proxy for quality of life, especially in dogs where you have voluntary euthanasia, when a dog's quality of life hits a certain low level. But then actually the other thing we're looking at is exactly this. It's explicitly quality of life. One thing is that frailty index, to your point. And the other thing is a dog owner reported measure of basically quantifying like how active does your dog seem, how happy does your dog seem, does your dog seem to recognize you, like things like that. And it's not perfect, it's not a great tool, but it's the best tool we have, and we've shown that it does correlate with a dog's disease burden, and it does correlate with some of the things that we're looking at mechanistically. So yeah, I think it's extremely important. I think honestly dog owners would buy something that just extended the quality of life, even if it didn't change the maximal lifespan at all.
Yeah, I really, there's something so lovely about this. Oh, sorry, did you want to go ahead?
Can I speak to that too?
Of course.
Because you all know about this demographic inversion that we have. We have The
Yeah.
ratio of elderly people to younger people is going up, up, up. And so that creates lots of problems, as you know. And one thing that would be great from a societal perspective is if these older people are healthier, if they're more functional.
Yeah.
And from a personal perspective of each person, obviously also to have a better, what we call a health span, is a real goal. And these changes that we're talking about that make people more resilient, they also make them disease resistant.
Yeah.
So I think that's a real, it's a real goal that we have. Yeah, and actually now I'm old. I wasn't. When I started, I was in my 30s. But I've been giving these talks, but I'm now old and I'm experiencing it. And this is Cynthia saying this. It could be I could be the only one that thinks this, but I am perfectly, I know, I maybe I'm wrong, but of course I know I'm going to die one day. One day that will be the last day. But when you're older, at least when I'm older, like I am now, I'm not thinking about dying. That will happen one day. But every day, you think, well, if I pick this heavy thing up, am I going to hurt my wrist? Or I get out of bed, a little creaky. You notice that stuff, and you don't like it. That's, I don't know about you guys, but that's what I think about, is just being able to do stuff I used to do.
Yeah.
Yeah.
Yeah, and well, so there's something so lovely about the idea of our dogs going out ahead of us on this kind of cultural experiment of living longer. And the science of this is sort of endlessly fascinating, but some of the cultural dimensions of it I think are really fascinating too. And I want to talk about those maybe in humans in a bit, but first let's talk about them with dogs. Again, imagining wild successes for Loyal, all your SF dreams coming true.
And Texas dreams.
And Texas dreams,
I'm an awesome
excuse me. Imagine that it were possible to double the lifespan of a dog, for instance. As we have in the roundworm. How would that change the shape of our relationships with these animals, which are deep and profound, but are usually circumscribed by kind of only occupying one particular stage of our lives?
Yeah. Well, so there's a couple of things to this, and this was actually the other portion of kind of the ideas that became Loyal that convinced me to start it, which was, there's always kind of been this negative narrative around working on aging and working on lifespan extension, working on longevity, right? If you think about it, it's been that way for centuries, right? It's these snake oil things that if you buy this, you'll live longer. If you rub this on you, you'll live longer. And it's just all BS money grabs, right? And now kind of the theme is billionaires trying to live forever, right? They have everything but longevity, and now they're trying to buy that. And one of the things I really love about dogs is that it's such a wholesome, very consensus idea to help dogs live a longer, healthy life. Even if you're a cat person, you believe that little angels on earth that we have, dogs, deserve to have a longer, healthier life. And it's just, it's not as fraught with all this socioeconomic and other, health insurance system issues that we have in the US, right? Now something that we worked on, I think we're gonna be able to deliver on is a drug that is priced at, in a way that the majority of Americans will be able to afford it from day one, and that was super important to me. So that was one. And I'm hoping that will help introduce this idea of longevity or aging, which I think is just a really sexy way of saying preventative medicine for multiple age-related diseases at the same time. That's a mouthful. Dog longevity sounds much cooler, so I say dog longevity. So I'm hoping that it will normalize it to the point where, there's a couple of different ways to affect change. I know there's a lot of political panels and whatnot going on. One is you can tackle it directly with lobbying or cash or whatever, right? The other is just kind of normalizing things in a cultural level. And I think if you can normalize the fact that I can go to the vet and ask them for a longevity drug for my dog, why can't I do that for my grandma? I think that's actually one of the biggest levers for enacting the sort of changes that will be necessary to make this start happening in humans. And then the other side of it was this idea of kind of building a pharma brand that people love. Because I think actually, obviously, there's a lot of issues with pharmaceuticals and drug availability and access and affordability in the US, don't get me wrong, but developing medicines is an extremely important piece of work. And it's shocking how low NPS a pharma company is, and I think there's an opportunity. If you're building a drug to keep your healthy dog, relatively healthy dog, healthy longer, and you're not being going broke or going into bankruptcy to afford it, and it's a positive thing, right? And it's all about celebrating the love that you have with this animal. again, I'm hoping that will bleed into an interest in the other things that are going on.
Let's talk about those other things that are going on. Cynthia, you come to us from Calico, an alphabet company, and Celine was telling us that basically, it's very difficult to do human trials because we live so long and maybe there's more red tape around it. Has that been really limiting for the ecosystem, or are we going to have our dog moment soon?
Well, yeah, lots to say about this. Lots and lots and lots. So I also want to make the point before I even start that all the work I told you about with these little worms was done at the University of California, which is a university, and it was funded by NIH, is basic research, because I was curious, and not just me, but all these people out there, they're just curious, can you do this? So I think this has created a whole industry that's real. It's not like there is snake oil there, but there's a real biology there. So I just wanted to give a shout out to the NIH for that. So that's the first thing. But then the next thing is, so we talked about this insulin IGF-1 pathway for aging, but there's a lot of other ones. Turns out, people of course have been working on this now molecularly for quite a long time, and there's all sorts of things that can make mice either live longer or at least live a lot healthier. So for example, you can take a young and an old mouse, you can stitch them together so they have one circulatory system. I know it sounds bizarre, but you can, anyway, or you can just inject old blood and, anyway, but the young mouse gets older.
This is the Peter Thiel blood boy thing.
Yeah.
The young mouse gets older and the old mouse gets younger. So you can try to find out what's in that. What is the factor that did that? Then there are certain cells called senescent cells that become more abundant with age that are like little inflammation factories. They're just spitting out inflammation. If you clear those out, you get a much better health span, happier old age. So I'm just saying that there's more than one thing. There even seem to be ways to roll back the clock, actually, which I won't talk about unless you ask me. But anyway.
I'm asking.
But, well, really?
Go right ahead.
Okay,
yeah.
okay.
Unless you guys don't want to hear about that.
I know, it's really cool. It's really cool. So you know about Dolly the sheep. You know that you can take a skin cell or a cell from an adult animal, or the nucleus from it, where the DNA is, and you can subject it to what are called Yamanaka factors, which are gene regulators, and they're named after the person who got the Nobel Prize for doing this. You can turn that old cell into an embryo cell, so you can basically fertilize it and have a whole new animal. So you can take an old skin cell and you can manipulate it with these Yamanaka factors, which are gene-changing factors. They turn other genes on and off. You can get back, all the way to the beginning. And then it was a skin cell, but now that one cell, when it's basically fertilized essentially, can make a whole animal, can make all the tissues. So that's amazing, right? It's like an extra pair of wings, maybe more amazing. But that was a Nobel Prize. People got their head around that. And it's called cloning, but you know that. So what's new and cool is it turns out that if you take those little Yamanaka factors and instead of keeping them going all the way back to the beginning, you just do it a little ways, in an animal, you can make the animal younger and you can make it live longer. I know, it sounds crazy, but you can do this. People have shown that they can extend the lifespan of mice that are kind of engineered to age too quickly, and they can make all the different tissues apparently younger. So it's tricky. We can't do it in people because, for example, one of these so-called factors also causes cancer, so it's called mix, so there's problems. But the concept is out there. The idea is that if we could kind of get control over these factors and make them obey us and do what we want them to, or maybe take some cells out, like old immune cells, and just kind of make them younger and put them back in. Maybe we could roll the clock back. So there are lots of people all over the place trying to see if that can happen. It's not as, this part, the IGF-1 sort of stuff, it just works. At least the mice, you just make the gene changes, they just all by themselves live longer. But this is different. We have to figure out how to do it safely. But, that's the way great things start, these kinds of observations. Anyway, so there are millions of companies now, not millions. There used to be at least 100. Now there's a downturn, so there are fewer than 100.
Okay.
Loyal exists, but there's still a lot of them, and there will be more again. There's an ebb and flow of these things. But what they have in common is that they're all science-based, and they're taking pieces of this biology that I just told you about and trying to make drugs out of it. And they're getting them approved first for diseases, probably, because there's a regulatory path for that, but then maybe for aging also, eventually. So can I say one more thing?
Yeah.
Oh, this is like, everybody has a hobby horse. Here's my hobby horse. So this is like a meeting full of famous people, some of who have a lot of money. And when I go to these meetings, I kind of feel like the camp nurse sometimes, because all these people that I think are amazing will come up and talk to me. I think, I really want to know how you make trucks, and they'll say, well, Cynthia. I said, what? And they'll say, I'm taking metformin, rapamycin, this NAD precursor, and taurine. Is that the right combination? What do you take? Should I be taking that? And I mean, everyone asks me that question, and they should ask me. That's why I'm invited to these things. But the fact is, I don't know what they should take. Number one, I don't know. And number two, this is really heartbreaking. I will never know. And the reason is that all those things I just mentioned are easy to get. So you can't have a phase 3 trial in humans that has, it's cost $500 million to $1 billion to do this right. This is like, even if we really knew how to measure aging, but even if you measure mortality or something that you can measure, these things don't cost very much. So you can't afford a trial. Forget profit. I'm just talking about getting your money back. Because let's suppose aspirin actually turns out to extend mouse lifespan a little bit. So let's just use that as an example. Let's suppose you find out that aspirin, this is who knows, I'm not saying it's true, but let's suppose it could extend your life, make you more resilient, and all that. What are you going to do? Compete with Bayer? I mean, how are you going to do this? So there's not a business model. But some of those things might work. They might really work. And we'll never know. So I, as a side project having nothing to do with Calico, would like to start a kind of what I call the World Health Span Organization. So it's like the World Health Organization except for health span. So it would be funded by governments, maybe nonprofits, and it would be a place that would fund high quality, large studies of all these things that extend lifespan in animals and seem good and are cheap and easy to get, so that people would know. Because if they work in people, I mean, everyone could take them. They're not expensive.
Yeah.
Third world people, everybody would be more resilient, have it, easier to support as an old person. It would be fabulous. But we can't do it because there's no business model. So just thought I wanted to say that.
No, I'm in favor.
I heard a clap. Thank you.
I want to give our audience a chance to ask some questions. I think we have a mic stand set up somewhere over here. And unfortunately, it requires traveling to the mic stand. We've got a volunteer. And we've got time for a few. Here's our first one.
Well, first of all, I'd like to know, are you thinking of trying any of these things on yourselves to expand your lifespans? But I read about in our newspaper in Sarasota County, Florida, that there was a veterinarian that used your dog, and it brought these dogs back to a healthy life, and it was wonderful. And I'm thinking, well, how can I get this for my son's dog who's old? And how long before we actually can? He'll probably die before he can get it. But how long before we can actually be able to have access to this helpful thing for our dogs?
Yeah, so we have these study sites across the U.S. We actually have one in Saratoga and quite a few in Florida in general. Lots of dog lovers in Florida. There's a couple of options. So you can enroll in the study if your dog qualifies, but the one thing you really should be aware of is there's a 50-50 chance of getting the actual drug or a placebo, right? Because we have to prove the counterfactual. Does a drug extend lifespan relative to a placebo? So that's something I try to be really candidly, if this, something that keeps me up at night is this study not working, not because the drug doesn't work, but because people are too worried that they have the placebo and elect to just go buy the drug when it's available. And so we've really selected for people who are excited and motivated by we cover all the vet care and the dog gets chronic care for the rest of their lives, but also who kind of want to be a part of history in some ways by participating in this study. And I think that's probably some of the stories you've read are from dog owners who feel that way. But we're running, and none of these things are guaranteed. Nothing is guaranteed. Asterisk, asterisk. I know I'm in DC. I promise I won't cross the line. But we're running towards hopefully conditional market approval next year. So that would mean that you could go to your veterinarian, and if they think it's appropriate for your dog, prescribe it. So. Fingers crossed.
All right, just a quick question. Celine, this is for you. I'm reading the statement that from the data that you collected, the FDA believes that the drug is likely to be effective.
Mm-hmm.
They rarely make this kind of statements. They usually say it's either effective or not effective. What does it mean, likely to be effective?
Yeah, so this is a really interesting. So one of the other kind of why nows for Loyal was this creation of this new regulatory path in the veterinary FDA called expanded conditional approval, which basically means that you have to hit the full normal safety bar, the full normal manufacturing bar, but only show reasonable expectation of efficacy. And then run your full study that proves definitive efficacy, or substantial evidence of efficacy is the technical term, which is this lifespan extension study. And they created this regulatory pathway in 2019 to basically make it commercially feasible to develop drugs that have really complicated and long pivotal studies, of which a lifespan drug is probably the longest pivotal study that you're going to get in dogs. And so what you're referencing is probably the most important milestone Loyal has had, which was us earning that, basically completing that effectiveness package through those pathways. So that reasonable expectation of effectiveness, FDA agreed that our drug that's based off of Cynthia's work is reasonably to be expected to extend the lifespan of dogs, aka we've hit the efficacy bar necessary. That's a huge deal. Most drugs fail, you know, and again, we haven't shown definitive lifespan extension, but we've shown quite a bit. But it was also the first time, as far as I'm aware, that the agency has publicly acknowledged that a drug can be approved for lifespan and healthspan extension. There is no disease, right? We're not developing drugs for any disease state. The inclusion criteria, there's obviously some niche exclusions, but generally speaking, it's by weight and age of your dog, and that's it. And that was one of the reasons why we started with the biology that Cynthia developed, because it's so broad, it's so broadly applicable, and that was something that was really important we wanted to prove that, right? And prove that a drug could be approved for lifespan and healthspan extension on its own. So yeah, hopefully more of those coming in the next couple of years.
Celine, Cynthia, thanks for being here today. This is really great.
Thank you. Thanks for having us.
For a conversation produced by our underwriter, Eli Lilly and Company, about reimagining Alzheimer's disease, transformations in care, please welcome Anne E. White, Executive Vice President and President of Lilly Neuroscience at Eli Lilly and Company, with The Atlantic's Nicholas Thompson.
Hello, Anne. Welcome. Thank you for being here.
Great. Very glad to be here.
You are working on some extremely interesting science. But before we get into the really detailed stuff and everybody blanks out, let's start with your personal connection to this issue, which is pretty intense and pretty important. Tell me why you are so committed to Alzheimer's research and how you got into it.
Well, thanks, Nick. And it is an exciting time. And I got into Alzheimer's research because, probably like many people in the audience, I lost my mom to Alzheimer's disease. But I lost her it's almost 20 years ago now. There's some pictures of her on the screen. She was an incredible mom. I would say she was even a more incredible grandma. And she would probably tell you that was actually her favorite role in life, was being a grandma. But if you think about her diagnosis and disease journey about 20 years ago, it was very different. There were not tests to confirm whether she had Alzheimer's disease until after she died. The only way to know that was through autopsy. And then there were certainly no medicines to offer at this point. And we're in a very different position today. So when I got the chance to lead this part of our organization, you can imagine I jumped in with both feet. And since then, have heard so many stories from different people about how they've been impacted by this disease as well.
And so Lily, you had been working at Lilly on oncology,
Yes.
and then Lilly had a breakthrough in Alzheimer's,
Yes.
and they said, we need somebody to run this research. All right, so now you have 2 drugs out on the market. How do they work?
Yes, so Lilly has one of the medicines that are out there, and then another company out there has another one. They both are targeting a plaque called amyloid. And so the way that Alzheimer's disease forms, essentially, is what happens is all of us produce amyloid naturally in our body. It's a natural protein that people produce. But what happens where things go wrong is when, essentially, these amyloids start to clump together and they form plaques. And when these plaques get to a certain level, they really start to cause really memory and thinking disorders when they get to that certain point. And so how these medicines work, they essentially attack that amyloid plaque and work to remove it from your brain, and that can meaningfully slow the disease.
Wait, so they stop the amyloids from joining together, or once the amyloids have joined together to create the plaque, they remove the plaque, or they stop the amyloids from forming in the first place?
You should be in research and all the different hypotheses that you'd like to put out there, because
If everybody
working
leaves
on all
the
of
Atlantic
those.
Festival knowing how these amyloids work, we will be in good shape.
Yes. So what these medicines do is they focus on attacking that amyloid plaque that has already formed and removing it from your brain. So that's the goal is people have formed this plaque. We want to remove it because as that plaque continues to grow, that's what leads to some of these
And
memory
how do they do
things.
that?
So essentially what it does is it causes, it's a targeted medicine, so you've heard about targeted medicine. So they target that amyloid, they attach to it, then they essentially stimulate your immune system to clear it out. So that's the way that these medicines work, is to really work together in partnership with your immune system to clear out the amyloid plaques.
So it's like they put like a garbage bag over the trash in my apartment, and then they like hire somebody to come and remove it.
Well, there you go, that's a great analogy.
So does that actually make the Alzheimer's go away?
So the challenge with Alzheimer's disease is that you will continue to form new plaques as you age and progress in this disease. And eventually it leads to a pathology called tau and tau tangles. And that also is what causes a lot of the issues, the symptoms that you see with Alzheimer's disease. So what these medicines do is they really meaningfully slow the progression. They don't stop the disease. Now, what we are working on is obviously moving earlier in disease, because what we know with these medicines is the earlier that you attack these plaques at the lower levels, you can have more impact, certainly. And so many of us have seen this across the studies that we've done. So moving earlier, I think we hope that we can even avoid many people getting some of these symptoms. But right now what they do is they slow that progression in a very meaningful way. And just to give you an example for my mom, she, in the picture that we had on screen, when she was helping care for my kids when they were toddlers, she had MCI. She was a very active person, able to care for her grandchildren in a way that was very meaningful. And if she could have had more time in that stage, and that's what these medicines had the chance to do, that would have meant the world to her. Another year, another 2 years with her grandchildren would have been very meaningful. So that's what we're able to do right now, and with insights and innovation, and you build on that and see if you can do even more.
Wait, so is the frontier figuring out a way to make diagnosis earlier so you can start on existing drugs, or is the frontier figuring out new drugs that can somehow treat the plaques or the amyloids at an earlier stage?
All of the above.
All of the above.
That's what we're trying to do.
Okay, but so then if they can start reducing these plaques as soon as you get them, what age do you start, if you're going to develop Alzheimer's, what age do you start getting these plaques?
Yeah, it depends. I mean, unfortunately for my mom, she started having symptoms in her late 50s. So that was a fairly early onset. The chances of developing symptoms, developing Alzheimer's disease, does grow as you age. So it's definitely a disease that becomes more prevalent as you get older. But it does affect people in their 50s, sometimes, unfortunately, in their 40s. So brain health is an incredibly important part of being aware of your symptoms, talking to your doctor.
So how do you test? How do you know?
Yeah, well, that's where I think there's been such tremendous progress. As I said, when my mom was diagnosed, it was all through just basically clinical testing. And that's certainly part of the diagnosis. But the other part is that we now have PET scans called amyloid PET scans that can actually really define if you have these amyloid plaques in your brain. And then really exciting is the emerging space of blood tests. So you can actually get a blood test to see if you have indications of this pathology in your brain. Yes, there are very good blood tests.
So we don't actually have to set up scans outside. We can just do blood tests outside for
we're
everybody?
seeing, I think the blood tests are still emerging, so there's still work to be done. But I think that Physicians are probably more used to using the PET scans today, so this is something that we and others need to do, is make people aware that there are blood tests that can also be used to diagnose that. So I think you're going to see blood tests continue to be used more and more as time goes on.
All right, so explain what you think the most exciting breakthrough will be next.
I think it is moving earlier in the disease. So there are a number of us doing studies in this space on patients, as I said, that don't have any of the symptoms yet of Alzheimer's disease. Because you think of Alzheimer's disease as the symptoms, but actually it's pathology. And if everyone could walk away with that thought in their brain, that this is actually a disease of pathology in your brain, it's not mental, weakness or, those types of things. It's very much about pathology, just like cancer is a pathology in your body. Alzheimer's is a pathology in your brain. And so if we can start detecting it and treating it before you have symptoms, obviously we hope that we can slow its progression, maybe even avoid, as I said, some people.
But how would you know? How would you know you have it before you have symptoms?
Well, that's a great question, because the fact is people are not walking around knowing this, so that's where we think that the potential of blood tests could come in. Yeah.
Oh, so you would be part of like a standard blood test. Right?
The hope would be at one point that you actually start to see this as part of routine screening.
I get my iron tested and I get my...
Yeah, just as easy. You get our cholesterol tested, you get your blood pressure checked, that you have this as part of the routine. But there's a lot of work between where we're at today and where that becomes routine. So
okay, so that happens, and then is it just like a stronger dose, a different dose, or are you doing different science to stop the disease at the early stages?
At that stage, it's still the same medicines, and we have other medicines in our pipeline as well that work on that, but it's still about attacking the amyloid at that stage. So it's still about clearing out and removing that amyloid at that stage. So it's these medicines, but in a different patient population, people that don't have symptoms, because the medicines right now are indicated for people that have the symptoms.
What is like the biological justification for amyloid? First of all, what is an amyloid, and what is the biological justification for an amyloid?
Well, this is a somewhat debated topic, and I would say that if you want to talk about the frontier, that's like a few years away.
I do, yes.
Which is understanding why do some people form amyloid plaques and others do not. And I wish I had the answer for you, and probably there's people in the audience that have some theories, but that is still a topic that I think we're all working on, which is how do you actually understand and maybe even change what's happening at the very beginning of this disease so that people don't form those plaques? But I don't have an answer for you today.
There's not like a biological trade-off. There's not a reason why these things exist. They're just wholly bad things that appear in our brains.
Well, as I said, they're a natural protein in your body, but what the bad part is when they start to really clump together. And so why in some people that happens and why it doesn't in others is still a scientific area that we're working on. But amyloid itself, as it naturally produces in the body, is not necessarily the problem. It's a problem when it starts, as I said, gathering together and then kind of sitting in your brain and affecting
all right, so this is super interesting. So if I were an executive, so explain it. This is kind of how your job works. You're there and you're thinking, okay, this is wonderful. We got to get out there. We got to market the drugs. We need people to use the drugs. We need the doctors to use the drugs. That's great. But the super interesting part of your job must be, I'm going to put $10 million or $100 million or however much money you have in your drawer at Lilly into one area of research and not another one. How do you make that decision, and what are you looking at right now?
Yeah. Well, I think that one thing I'm most proud of about Lilly, many, many things I'm proud of, one of them is that we have been working on Alzheimer's disease for 35 years. So it's been an incredible journey. And a lot of people say, well, don't you feel really lucky that we finally found an answer? But it wasn't luck. It was like learning from every single experiment and never giving up.
Right.
So the fact that we've continued to invest in this space makes me extremely proud. And the one thing we want to do is always say, how do we build on that insight that we got now that we have a medicine that treats early symptomatic Alzheimer's disease, is how do you make that result even stronger? How do you go earlier? How do you prevent the disease from progressing? So that's a big part of it. There are other things that we're attacking too. Probably the other exciting thing to note, though, is that the insights that we have here could play out in other really hard to treat brain diseases as well. And so we're definitely looking into
because you don't only have expert in clearing amyloid plaques, you have experts in how to
attack things in the brain. Yep, how does the brain respond to these types of things? How do you manage these medicines? All of that's going to play into this.
Can you make me better at learning languages? It's really frustrating.
I think like most things we were talking about, the earlier you start, the better. So it might be a little late for you and I don't know. Sorry.
Yeah.
So I took many years of Spanish, can't speak a single word of it, basically. So,
but the frontiers of brain health are super interesting because so much of what happens as we get older, many of the disease are mysterious things happening to the brain. So the idea is the Alzheimer's work could not only fix Alzheimer's, but could potentially help with
Parkinson's, ALS, MS, some of the very hardest to treat diseases, insights that we get in any area in neuroscience. I mean, this field is incredibly collaborative. The work goes out there. We work together and figure out, okay, how do you take this insight and then build the next one? And that's what we did in Alzheimer's disease. This was insights that came from the field, from academia, from pharma, all working together to make sure that we shared every insight from every study, figured out how to design the next study, which patient population, which target, how much to give, all those kind of things were things that we learned together to get here, and then we'll do the same for Parkinson's, ALS, and those types of things. So that gets me really excited. I would say the other area that I spend a lot of time on, and it also excites me, is we've got great medicines, but they're not reaching enough people.
Yeah.
So a medicine can be wonderful, but if nobody receives it, then it's not going to make a difference for that patient population. And Alzheimer's disease, unfortunately, is extremely underdiagnosed today. The fact is, most people, on average, can take about 2 years before they actually get that diagnosis. And that was certainly the case for my mom. We went through a lot, strokes, other things being ruled out before we finally figured it out. Only about 20% of the people with early symptomatic Alzheimer's disease are actually diagnosed today. So I get excited about the science, and that's certainly the future as well. But right now, I also spend a lot of time working with physicians and healthcare systems on how do they make sure that they're able to help the millions of people today that need help right now, that could benefit from these medicines but are not getting them because we don't have enough neurologists, people aren't using diagnostic tools regularly, people aren't talking to their doctor. I mean, my own mom, she wouldn't tell anyone about her diagnosis forever. You know, the stigma was overwhelming. And if we can start saying this is a disease of pathology, this is not a weakness, this is something just like cancer that you need to speak about, get support on. That will feel like a legacy for me as well, as if we can change that quite a bit and get more people to actually benefit today that are suffering today.
Yeah. Well, that is a great message. You're doing remarkable work. Thank you so much.
Thank you.
All.
Great to be here.
That was wonderful.
We want to thank Eli Lilly and Company for their support of the Atlantic's journalism. Please refer to the festival agenda for today's complete schedule. We hope you enjoy your festival experience. If you are attending